Amsterdam, July 1992. AIDS activists from around the world have converged on this picturesque European city to stage a series of demonstrations aimed at highlighting a whole range of issues that affect the lives of people with AIDS, including discriminatory immigration policies, the slow pace of treatment research, media misrepresentation, unethical clinical trials, and misdirected education programs. Over the years, AIDS activism has carved a unique place in the history of political movements. Never before have a group of people affected by a common disease banded together in a way that transcends boundaries of class, race, gender, and sexual orientation. Their distinctive blend of street theater, activist artwork, cultural criticism, coupled with media savvy, have allowed AIDS activists to achieve some remarkable goals in just a few years. They have managed to effect fundamental changes in the way new experimental drugs are tested and released, not just for AIDS, but for all terminal illnesses. But they're still faced with an age-old legacy of homophobia, racism, and sexism that in many ways present as great a challenge as the illness itself. And today in the streets of Amsterdam, they are determined to make their voices heard in one of the most colorful and forceful demonstrations in the movement's history. Carrying black cardboard coffins to symbolize the lives of those who have died, the AIDS activists try to enter the lobby of the RAI Congress Center, which is the site of the 8th International Conference on AIDS. But they don't manage to get very far.
Bent u gek geworden? Ik heb hier een lidmaatschapskaartje. Ik mag erin. Ik mag hierin. Ik heb hier toestemming om erin te komen. Ik mag hierin. Kun je nou op deze perskaart? Ik mag hierin. Laat me niet meneer. Ik heb een kaart. Dat is een regel de tas. Dat zijn regels. Hier mag de pers ook naar binnen, meneer. Dat is helemaal goed. Kaars komen er niet eens naar binnen. Nee, je kan dus met een kaart niet naar binnen komen. Dat is ook een schande.
What do we want? The cure for AIDS? When do we want it? Now!
Inside the conference halls, away from the angry shouts and screams of the activists, scientists sample the latest findings from around the world in a maze of aisles full of thousands upon thousands of posters dealing with every conceivable aspect of the problem, from safe-sex campaigns to AIDS treatments. These annual affairs have become so big, it's difficult to know where to start, and many people just simply use the time to catch up with old friends. AIDS groups from every continent are here, setting up their displays, each with their own distinctive approach to the problem. Corporations, large and small, are here too, peddling their wares in what's been dubbed the Disneyland of the AIDS industry. At the heart of all this activity lies a tiny piece of genetic material, the retrovirus HIV. Almost all AIDS research has been focused on HIV since it was first discovered 10 years ago. But nagging questions remain. In vitro test tube observations don't necessarily apply to behavior in vivo in the body. Only a tiny fraction of T cells in the blood are actively infected with HIV. There is no proven mechanism by which HIV causes AIDS. There is no applicable animal model in which HIV causes AIDS. Various cofactors seem to play a significant role in AIDS, and there is no correlation between actual viral activity and disease progression. But despite these problems, HIV has remained the object of almost all AIDS research, since the syndrome is nearly always accompanied by antibodies to the virus. That's an association that even one of the most fiercest critics of HIV, Dr. Joseph Sonnabend, acknowledges.
One cannot deny an association. There is an association, even if it's not 100%, it's certainly overwhelming. Now, you can have an association for more. Association itself doesn't imply cause, and it's my belief that an alternative view is actually much more likely. And this alternative view is that the appearance of antibodies in a person who will go on to get AIDS or may not get AIDS is really a consequence of some immunosuppressive condition or which I'll come to, rather than a cause of it, not or associated with the cause. Antibodies don't cause anything, but rather than be associated with the cause, the appearance of antibodies are a consequence of the conditions associated with the true causes of AIDS. And I should explain, try and explain this. Following infection with HIV, people don't make antibodies unless the inoculum is massive. Antibodies are made to protein, and the protein is in the core and in the coat of the virus. If you get a small inoculum of the virus, there's not enough. There may not be. I'm suggesting there isn't enough protein there to elicit the body to make an antibody response. But the virus, what happens is the virus becomes latent. It loses its protein coat, which is destroyed. The DNA, which carries the instructions to make more of that protein, is plugged into the cell, and that's where it remains. And unless that is activated, you're not going to make antibodies.
And in fact, a growing body of evidence shows that people can harbor HIV, but without making antibodies. A variety of techniques, including analyzing the genetic sequence or genome of cells, can now find HIV in people without HIV antibody, many who've remained this way for years without activating the virus.
A lot has become known about what activates the genome to make antibodies. There are a whole lot of things that are now understood that will turn that genome on out of latency. For example, there are what are called some of the cytokines. For example, a substance called tumor necrosis factor, which appears during the course of a variety of infections. If it's there, that in the test tube can activate HIV out of latency, for an example. And certainly people with AIDS have lots of TNF, tumor necrosis factor, in their blood. That occurs in things like malaria, it occurs in drug addicts. It's a part of the response to an infectious process, and so that is able to turn on HIV. Possibly some of the other cytokines are able to do it too, and these might appear under conditions where people are infected with other agents. This may be associated with producing these cytokines, which in turn will turn on HIV, which in turn will make you'll make virus, and therefore you'll seroconvert and become antibody positive. Now, other viruses can activate HIV. One knows this. For example, if you infect a cell with the HIV-infected cell is super-infected with cytomegalovirus, herpes virus type 6, a variety of viruses. The super-infecting virus will turn on the latent HIV. But that requires that the same cell is infected with two viruses. I guess that may happen, although I don't think that probably happens too often, but you could imagine a person carrying latent HIV who then becomes infected, the same cell becomes infected with CMV, that cell will then start making HIV. It will turn on. So that's another potential mechanism for turning on a latent virus that's come to be understood somewhat. Another possibility is that the immune system keeps HIV in check, that's to say keeps the virus latent, and which would say that one gets infected with a small inoculum with HIV, the HIV integrates into the genome, that's the end of the story. If you have a healthy immune system and these other conditions don't operate, like the TNF, the other infections, but one could imagine that I'm saying imagine, this has been demonstrated for other viruses that are latent, these mechanisms I'm about to speak about. When a cell starts to produce HIV, then cytotoxic T cells, other cells of the immune system, recognize this and kill that cell before it's produced too much virus. And in that way is yet another mechanism for keeping HIV in check. That's been worked out for the Epstein-Barr virus model in some detail. So and these cytotoxic cells that will kill HIV-infected cells, actively infected cells, have also been described already. So it's very likely that this exists. Now, if this mechanism of killing is perturbed by some other immunosuppressive influence, this will permit HIV that's coming out, there's a second line of attack, as it were, that's been induced, will continue to be produced. And so immunosuppressive conditions may promote seroconversion for the reason that the immune system doesn't keep the latent virus latent. It allows latent virus, permits activation of latent virus to just proceed.
At the Amsterdam AIDS conference, researchers from the U.S. National Cancer Institute presented evidence showing that 2/3 of HIV antibody-negative gay men that they tested had in fact made a T-cell response against HIV, but without making antibodies. This rather astonishing finding was reported in the London Sunday Times by Neville Hodgkinson, but hardly anyone else seems to have noticed. Other scientists, like Jonas Salk, think that this T-cell activity is the first step of the body's immune response against HIV, which may actually keep the virus in check, unless the cellular arm of the immune system is somehow disrupted. Coupled with this is the finding by a group at St. Paul's Hospital in Vancouver, showing that a prior defect in cellular immunity was a better predictor than risk behavior itself for developing antibodies to HIV. They wrote, "An impaired host immune status may be associated with an increased risk of HIV infection that is independent of risk of exposure to the virus, supporting earlier speculations that HIV may itself be opportunistic."
When HIV was first proposed in 1984 as the cause of AIDS, people like Jay Levy in San Francisco asked, well, how do we know that HIV, since it's a latent virus with a latency mechanism, how do we know that it's not merely a reactivation as a result of whatever is really causing AIDS? In other words, how do we know that HIV itself is not merely an opportunistic complication of whatever is really causing AIDS? No one ever answered that question, why it isn't just as likely that you can account for the presence of HIV as an opportunistic reactivation instead of calling it the cause.
So we have conditions then that promote seroconversion, which are quite different to the conditions that are associated with infection, although they may overlap. Or yet another way of turning on HIV, I should say, is what are called alloantigens, which is foreign antigens. The usual way of isolating HIV from lymphocytes is to mix the lymphocytes containing latent HIV with cells from another person. And that sets up a mixed lymphocyte culture. That's the usual way. It's called co-cultivation. It's the usual way of doing it. And the reason how that works is that for some reason the cells carrying latent HIV recognize the foreign cells in a particular kind of fashion, and this the end result of all of this is the cells are stimulated by this foreign cell, and in the process they will start producing HIV. Now, people with AIDS are certainly exposed to alloantigens, whether it's in blood in people who share needles or through semen in the case of homosexual men. We have to assume that rectal deposition of semen is somewhat different to the vaginal exposure, and that's not so way out because the epithelium is different, and the lining is different, and it's not unlikely that alloantigens get into the body through the rectum, and as such they could activate cells containing latent HIV. So here's yet another mechanism which dissociates seroconversion from infection.
Lending support to this idea is a remarkable experiment carried out by Tracy Keon and Jeffrey Hoffman, researchers at the University of British Columbia. They injected healthy mice with white blood cells from other healthy mice and found that the injected mice began producing antibodies against HIV. Hoffman suggests this raises the question over whether exposure to foreign cells in itself may cause HIV seroconversion, regardless of whether those foreign cells contain HIV.
Clearly, it seems to me a more intelligent way to look at the possibility of infection is to do the following, is to say that the risks for infection should be looked at differently than the risks of seroconversion. They're quite different events, and that infection may well be casually. Maybe we don't know this. We just assume that you can't get HIV by casual contact. However, you can't get AIDS that way, but it's not entirely. You may well get HIV that way if you dissociate the two things. The two things being infection and seroconversion as being rather distinct events. So infection is it gives you the virus. The risks for seroconversion are to be found at least in the things I mentioned, whether it's cytokines, whether it's infection with dual infection with another virus, or whether it's alloantigens, and who knows, or immune suppression. There are at least 4 different things that are known to activate HIV out of latency. So an individual may be infected is one step, no antibodies, and then 30 years later they may receive a blood transfusion, be exposed to alloantigens, and that would be sufficient to activate their HIV that has just been dormant for all this time somewhere. And so it would be much more useful to dissociate these 2 events. Now, if you do that, then it says that the association of antibodies with AIDS may result from the fact that the conditions that produce AIDS also activate HIV out of latency. And there's no argument against this yet. And the conditions that produce AIDS may be those associated with abnormal production of cytokines. We know that people with AIDS have TNF, they have IL-1, they have stuff in their blood that people with AIDS, also, without AIDS also some of them have, but certainly if people with AIDS have these problems, they certainly carry multiple herpes viruses for the most part, which could transactivate HIV. They have immune suppression, whatever's causing it, which could allow HIV to be activated, and they're exposed to alloantigens which allow HIV to be activated. So all this says that if in these other activating circumstances, or if these are associated with the true causes of AIDS, then HIV seroconversion in the presence of antibodies is a consequence of the true causes of AIDS rather than the cause of it.
If a variety of infections and other conditions can activate HIV out of latency, it may well mean that HIV isn't some new mysterious virus after all. In fact, genetic analysis by the Nobel-winning scientist Manfred Eigen has convinced him and people like Jonas Salk that HIV is probably hundreds of years old.
I don't believe HIV is new. One has to really ask what is meant by new when we're speaking about a new virus. What new means... The 2 most popularly accepted theories about what newness means is one that is new in human populations because it's come from another species, the green monkeys or whatever is supposed to be the fatal bite. Actually, nowadays we have to have 2 bites, but I'll come to that. The second one is that there's been a mutation in a virus that was harmless before, and that mutation turned it into a lethal virus. Problem with that one is that if there was a mutation, then there has to be a virus that existed before the harmless virus. We should surely find evidence for it. We've certainly found no such thing, so it's a very unlikely event. But even if it did occur, the definitive argument against this comes from the fact that there are 2 viruses that cause AIDS. There's HIV-1 and HIV-2. There's been an incredible silence from the scientific community on the problems that these 2 viruses pose. They are 2 different viruses. They are not the same virus. They may both be retroviruses, but more than 60% of their genome differs. So this makes them 2 different viruses, not just as is trying to be fobbed off 2 strains of the same virus. That just simply is not on. So if there was a mutation, one would have to say there were 2 mutations, and that 2 mutations should happen in 2 different viruses, causing both of them to produce a similar kind of illness is beyond what probability would permit. It's just totally not possible. We could conceivably have 1, but that 2 should happen in the same time frame in 1960 or 50 or whenever it was, that this particular mutation or mutation to lethality should separately occur in 2 different viruses is beyond anything that's reasonable. It just simply is not an argument that even can be entertained. If it's a question of a monkey bite, then we have to say that one monkey gave man HIV-1, another monkey, well, I think it's a mandrill another monkey in a different part of Africa gave people HIV-2, but at the same time. So all one can say maybe is that there was a frenzy of monkey biting that took place in 1950 that led monkeys to bite people all over Africa. A green monkey gave people HIV-1 during this frenzy in the middle of Africa, and somewhere in West Africa another kind of monkey gave people HIV-2 during this crazed monkey biting epidemic.
A great deal of foolishness was put forward. I mean, there were scientists speculating on little African children carrying around dead monkeys as pets. Well, this is ghastly. I mean, what child would choose to have as a plaything a dead animal? There was no evidence for it, but that, and then they thought that Africans, devoured monkeys, and that this might account for how the virus somehow passed from monkeys to human beings. All not just speculation, but particularly offensive and ridiculous speculation.
The only other argument that one might say is that AIDS existed in human populations in remote areas. In one area they were getting HIV-2, in another area they were getting HIV-1, and suddenly in 1950, with these remote tribes came into civilization. Well, that's also ridiculous, because Africa may be a dark continent and remote and all of that, but it is not that impenetrable, and for centuries there's been traffic throughout Africa, and the idea that there were two tribes totally kind of secluded, each with their own kind of AIDS caused by different viruses, but they would, and all of a sudden these tribes somehow penetrated into other areas at the same time is kind of unlikely too. So we have to entertain the fact that the HIVs, when we speak about HIV-1, the same thing would go for HIV-2, have been preserved in nature.
Millions of people worldwide have HIV, yet it's remarkably difficult to transmit, often requiring hundreds of sexual contacts. That's puzzled Toronto researcher John Scythes.
Its lack of infectiousness makes it very hard for me to understand why it appeared around the world at the same time. And it did, really. I mean, within a very few years, many communities all had HIV in their prostitutes or their gay males or their sexually active heterosexual populations, depending where you go, or in London or in Paris, in the gay male communities, etc. Did someone fly around and sprinkle this virus down on everybody? It's not infectious enough. I mean, maybe homosexuals jetted around a la Gaëtan Dugas, or whatever his name was, a la Randy Schultz, but the heterosexuals didn't to the same extent. And it's very hard to explain. It's rather a quick appearance unless it was already endogenous, i.e. harbored within the hosts. And their immune suppression from other causes allowed it to perhaps have clinical significance.
Now, this doesn't argue against the fact that once it's activated, it may indeed contribute to the disease process. I mean, it may indeed, but it cannot do so unless all these other conditions are met. And in an operational sense, I would say these other conditions become the cause of AIDS, rather than, even though HIV, once activated under unusual circumstances, may contribute. I mean, I think that may indeed be possible, but operationally, it's not the cause of AIDS. So it's the conditions that activate it are the unusual disease-causing conditions.
So HIV has clinical significance in prior disposed or immune-suppressed individuals. I think that may be a fair statement. HIV may prove to be pathogenic in ill people. After all, they do make antibody. So that's interesting and compelling in and of itself. But it doesn't prove chronic disease, and it does not prove pathogenesis, the presence of antibodies.
In fact, the antibodies made against HIV appear initially to be quite effective in suppressing the virus. This 1991 study found that after the appearance of HIV antibodies in the blood, the amount of an HIV protein called P24 antigen dropped to undetectable levels within 7 to 12 days. The authors wrote, "The rapid and spontaneous decline in the viral load suggests an effective immune response in the host. This is the case with most viral infections, in which antibodies are usually a sign of immunity, although there are some exceptions."
Antibodies usually are a sign of immunity, but antibodies are also a sign of chronic disease. So you have to, that's one you have to be careful of.
The presence of antibodies does not negate the fact that an organism may cause disease. Viruses can spread from a cell to cell. They don't need to be exposed to antibodies even. For example, shingles occurs in people who have antibodies to the virus that causes it. It won't occur in people who don't have antibodies. Herpes recurrences occur in people who have antibodies. It doesn't protect you from a recurrence. Cytomegalovirus infections, severe ones, doesn't help that you've got antibodies. You could have even a lethal infection. Antibodies, it makes no difference.
However, in these types of situations, the actual number of cells found to be actively infected with these viruses is far larger than with HIV, which can only be found to infect a tiny fraction of cells. This suggests that the antibody response results in effective immunity against the virus, as is usually the case with most other infections.
Antibodies usually, that's fair, usually are a sign of immunity. It's fair to say that, especially when they're very constant, like HIV. HIV antibody titer goes, I mean, you know how it works, and antigen goes whoosh, like that, and the patient takes care of the bug, and when that drops, the detectable antibody rises. This is the window period in the theory, 4, 8, 12 weeks, whatever, and then it stays rather constant for years until a patient becomes debilitated and goes away. It's suspiciously simple for such a complicated relationship. The collapse of the human immune system and things that happen along the way, like an acute PCP happens and the patient does very well for 2 years afterwards, but good old HIV titer stays right on steady, and yet it doesn't infect the cells. It's hard to take.
A lot of dogmatic assertions have been made about how HIV behaves, and these are just totally fanciful, and I'm sort of ashamed of medical scientists. Well, I shouldn't really say that, but it's I think the public has, and ordinary doctors and just people out, have been really let down by those who've taken on the interpretation of this disease. I recall some time ago that there was maybe about 4 or 5 years ago, there was a television program in which Dr. Fauci was telling the world how HIV caused AIDS and telling the world about the course of HIV infection. And he said that people get infected with HIV following a window of, his words, of seronegativity of 3 to 6 months, I think 3 months and maybe up to 6 months, seroconversion occurs, and there were pictures drawn of infection, and then 3 to 6 months, a line shows the antibodies coming, and then you see the P24 antigen goes away and then comes back sometime later as people. All of this is just totally fanciful. It's presented as if these were kind of a factual representation. It came from animal, I know where it comes from, it just comes from experimental infections of animals with a variety of other viruses. You cannot, HIV is a retrovirus, and there are no models in which to base this kind of authoritative picture of the course of infection, and that has done a tremendous amount of damage.
It is noticeable how unscientific the people in the establishment are. One of their claims was that they had cases of AIDS which had been caused by needle sticks, accidental jabs with a needle. And in fact, the claim was completely spurious on the basis of their own evidence. What happened actually was that health workers who suffered needle sticks of HIV-contaminated needles did suffer some positivity. But whether it's from the needle sticks or not is unproven. But anyway, about 17 out of the 2,000 who said they'd been stuck with needles came up with seropositivity, and only one person was counted as an AIDS case, as such, developed supposed AIDS symptoms.
A good workup has revealed that indeed the person had other risk factors: drugs, lifestyle, sexual behavior, etc. A partner who could easily have other infections, in other words, a high-risk partner for developing AIDS, HIV notwithstanding, an immune-suppressed person.
It turned out that was in fact the only supposed claim of AIDS that had resulted from needle sticks.
Needle sticks should work if it's as deadly as we say it is. There should be, you're not pushing on the plunger, but some virus gets in. The time that needle sticks do work, for instance, a dirty needle infects 6 babies or 10 babies in Russia, and the news hysteria around the world is rampant. But those babies are bubble babies. They're premature. Their immune system isn't even working. That's why they're getting all these transfusions. They are A-gamma globulin-emic infants with stem cell disorders that may or may not live, HIV notwithstanding. So always remember that when you find HIV antibodies in babies that are premature. What are these babies doing in the ward where they were getting these transfusions? Normal births don't get transfusions. Why don't people put things in the news the right way? So the people like me don't have to sit here and poke fun at them. But of course they get away with it. They're the news. And the doctors don't scream, and they should, because it's bad science. Those reports from Russia are okay to make, but qualify them. Why are those babies in an intensive care unit? And why do they become HIV positive from one needle, a whole bunch? Because they are prior immunodisposed.
The whole question of AIDS in infants has also been thoroughly investigated by Michigan State University professor Robert Scott Root-Bernstein. He's found that babies with AIDS have a number of other health risks besides simply HIV.
Many HIV proponents point to infant AIDS as probably the most important proof that HIV is the sole cause of AIDS. And the argument is very simply that infants don't have any lifestyle risks. What can they possibly do that would immunosuppress them? And that sounds at first glance as if it's a very impressive argument. But if you look into the data very carefully, you find out that it doesn't hold up at all. Something like 90%, maybe it's 85%, I'm not sure of the exact figures at this point, of infant AIDS patients are born to drug addicts. The infant itself is often born a drug addict. They are almost always born premature. They're microcephalic, that means they have a small head circumference, and their immune system is almost always impaired. Now, part of that is simply because they're premature and microcephalic. We knew long before AIDS that those are risk factors for immunosuppression in tiny infants. More often than not, the reason that they're born immature is malnutrition or the fact that the mother has multiple infections such as gonorrhea and syphilis and so forth and can't carry to term. The child will often have these infections or has to be treated for them. So when they are born, they'll be drug addicts, they'll be malnourished, they'll often need blood transfusions, they will have various infections they have to be treated for, and their immune system is not as developed as a full-term baby. Now, if that doesn't make you prey to all sorts of opportunistic diseases, I don't know what does. Then again, there are the other large group in the infants who get blood transfusions. You have to ask yourself, why does an infant get a blood transfusion? It's because that infant is deathly ill. There's something wrong with that infant when it was born in the first place. So in addition to getting the blood, which is immunosuppressive, in addition to getting antibiotics to treat whatever its surgical problem probably is, in addition to getting anesthetic, which anesthetics can be immunosuppressive, this baby was born very, very ill. I think that speaks for itself. They may not have lifestyle risks, but they certainly have immunosuppressive risks other than HIV.
And the same would appear to be true for all the other various groups of people at risk for AIDS, whether it's newborn babies, heterosexuals in Africa, IV drug users, blood transfusion cases, hemophiliacs, or multi-partner gay men. In each instance, a host of other factors are present that could not only activate an HIV infection, but could also contribute to many of the immune irregularities associated with AIDS. These other factors might also explain the tremendous diversity of symptoms among people with HIV antibody. These manifestations vary widely depending on the specific risk group, and some, like AIDS dementia and lymphoma, are not even traditionally associated with immune suppression per se. Professor Harry Rubin is one of the world's foremost experts on retroviruses, having studied them since the 1950s. Like his colleague Peter Duesberg, he finds it hard to believe that a relatively simple virus like HIV could cause all the different manifestations of AIDS.
I don't think the cause of AIDS has been found. I think in a disease as complex as AIDS that there are likely to be multiple causes. In fact, even to call it a single disease when there are so many multiple manifestations seems to me to be an oversimplification.
I think that in this area, Peter Duesberg has answered it very, very well in his paper in the Proceedings of the National Academy. He touched on the tendency of HIV, or at least the alleged ability of HIV, a tiny 9 kilobase virus. You understand it's a very small organism with no DNA of its own, to be able to cause risk group specific illness, encephalopathy in one group, wasting syndrome in another.
This is especially noticeable in Africa, where the diseases associated with AIDS are quite different than those in the US and Europe. All the same, it's claimed that HIV is what's responsible for the African immune suppression. But there well may be other factors.
If we look at Africa, we find that the places in which AIDS is a major problem are all places which are, one, extremely poor. They are the poorest countries in Africa. They are the ones that have the worst healthcare systems. And if we look at the disease load of the individual people, they have the highest disease load. And when I say disease load, what I'm talking about is the following. If a patient walks into a hospital, they will often have a number of viral infections, bacterial infections, fungal infections, various kinds of parasites, and so forth, in addition to HIV. So we're talking about a person who might have 8 or 10 chronic infections simultaneously. Malnutrition, protein-calorie malnutrition, vitamin A deficiencies, vitamin D deficiencies, and so forth. Iron anemia, iron deficiency anemia. These are all very, very common in these countries and very, very uncommon outside of third world nations. Now, all of these things are known to be immunosuppression. Put them all together, and HIV certainly may be a factor, but we already have a population that is very, very prone to all sorts of infections.
People have not exactly had the longest life expectancy, and I think, again, Peter's points are well taken there, that AIDS, or AIDS-like illness, was alive and well in Africa before the discovery of a human retrovirus.
And even since the identification of HIV, a significant number of people in Africa who fit the AIDS profile, in other words, previously healthy people who die prematurely of opportunistic infections not usually considered fatal, turn out not to have HIV. In this study from Ghana, presented in Amsterdam, an astonishing 59.5% of suspected AIDS cases tested negative for HIV, suggesting that HIV-free AIDS is considerably more widespread than we've been led to believe.
Another major argument for HIV, of course, is that hemophiliacs get AIDS. Again, the argument is made that only HIV is the immunosuppressive risk there. Again, if you look at the cases carefully, that isn't true. In most cases, hemophiliacs need transfusions several times during their lives. In fact, there's a very direct correlation between age and whether hemophiliacs develop AIDS. Younger patients or younger hemophiliacs tend not to develop AIDS. Older ones develop it much more quickly, which indicates there's something about getting older, the immune system being suppressed due to their disease, which is a factor here, and that could be transfusions. One thing that all hemophiliacs have to do is treat themselves with blood products, and anti-HLA problems have developed in almost all hemophiliacs. Even hemophiliacs that are HIV-free are known to have T4, T8 imbalances that are typical of AIDS patients and so forth.
It's not exactly healthy looking at HIV-negative hemophiliacs immunologically, which we have several references on. You cannot pick out which hemophiliac is HIV antibody positive, blinded from their HIV antibody status. You can't pick them out. In other words, you look at their immunologic parameters, and boy, these people have got wonked out T cells.
This 1989 study of HIV-positive and HIV-negative hemophiliacs found no differences in immune abnormalities between the two groups.
So again, hemophiliacs, when we look at even the healthy ones, are known to have immune problems, and they appear to be directly related to blood products.
At this 1992 AIDS conference, Luc Montagnier, whose team originally discovered HIV, also reported immune abnormalities in HIV-negative children receiving treatment for hemophilia.
So this tells us perhaps there are some common factors in hemophilic treatment. I believe this is like you do. This is due to the heavy protein treatments the hemophilic children got until recently, because now they are inoculated with highly purified factor 8 or 9, and this remove most of the contaminant proteins. But in 1980s, in 1985, they received concentrates of proteins which were 1% active factor and 99% contaminants, impurities. And those impurities stimulate in a bad way the immune system, and they get immune depressed some way. But this is very interesting that even now the hemophilic child which are seropositive, they do better when they receive purified factors.
In fact, this Italian study shows that even after 2 years' observation, HIV-positive hemophiliacs who receive a new, highly purified clotting factor concentrate show no significant decline in their T-cell count. 2/3 of them even regain the ability to react to various antigens in skin tests, which are a measure of immune function. On the other hand, those on intermediate purity showed a drop in T-cells, and some developed AIDS. This finding has been duplicated in a number of other studies, prompting some researchers to suggest that an agent or agents in older clotting factor other than HIV may be responsible for the immune defect. Immune dysfunction that's independent of HIV is also seen in other populations at risk for AIDS, including intravenous drug users, or IVDUs.
See, IVDUs don't look very good immunologically. Again, they're much like the hemophiliacs. You work them over with an immunologic workup. When they have HIV antibody, they're sicker and die. But when they don't have HIV antibody, they are not in very good shape. I got a paper from Courtney Bartholomew, went to Trinidad in 1987 and published a nice piece in the JAMA, which is a prime example of this, where you could not predict which of the gay males. He screened 110 gay males for HIV and various immunologic parameters, helpers, suppressors, inverted ratios, history of other STDs. The T cells were not significantly different at all. As a matter of fact, they were within 30. Whether the group was positive or negative, and there were people who were HIV negative with under 200 T cells. The mean was 200 to 2,000 in the negatives and 0 to 2,700 in the positives for HIV antibody, with an average in the mid-500s for both. In other words, HIV had no predictive value on immune suppression at all. The exact opposite of what's reported in our cohorts here, where they screen HIV antibody negative people and say, look at how healthy they are. Then they find the HIV antibody and look at these T cell levels. But someone else found the exact opposite in Trinidad. How do you explain that?
This discrepancy is only one of the many anomalies in the current HIV/AIDS model. Even in the presence of HIV antibody, the rate at which different populations develop AIDS differs substantially depending on the particular risk group, method of exposure, geographic location, age, and so forth. For example, in this comprehensive 5-year study of AIDS in Uganda, published in 1990, it was estimated that 1 million adults in that country were HIV positive. This is based on the fact that 42% of randomly selected patients from 8 hospitals tested positive for HIV in 1986. And yet, as of 1990, despite a highly successful surveillance and reporting system, Uganda had reported only 8,000 cases of AIDS. And although that number has risen since then, it's still only a fraction of the AIDS rate in the U.S., which has reported over 200,000 cases, among a similar pool of 1 million HIV positives. These figures suggest that the development of AIDS may have more to do with other factors than merely HIV, and that a substantial proportion of HIV-positive people may never get sick.
Yes, and we are seeing 30-year free of AIDS risk groups. I've got the papers with HIV antibody. The Johns Hopkins study just said 13.6 years, which was very surprising, and the Vancouver St. Paul's people, the lymphadenopathy study, identified a low-risk zero-incident group of HIV antibody positives with a predicted 25 to 30 years mean time free of AIDS.
That study, looking at the T cells of low-risk gay men with known dates of seroconversion, estimated only 13% would develop AIDS within 5 years, and taking other immune abnormalities into account, the rate dropped to 6.7%, implying a mean time free of AIDS of over 35 years, in sharp contrast with earlier projections. The researchers suggested these other immune abnormalities may be more important than simply T cell counts in predicting who gets sick. In hemophiliacs, prior immune suppression seems a better predictor of AIDS than just having HIV.
If you study the hemophilia literature, you will find as many papers that show prior immune suppression, and you will find papers which show that you cannot pick who is HIV positive by the immunologic parameters. You will find as many papers that way as you will find that support a progression of 5, 8, 9 years to AIDS in big in-city hospitals. So the question is far from answered. It's very much up in the air.
The commonly cited progression rate of 10 years from becoming HIV positive to developing AIDS is based on figures that include those who've already died in the early part of the epidemic, before the introduction of safe sex. But for the last few years, the number of newly diagnosed AIDS cases in Canada has leveled off and is now starting to go down. Yet if HIV causes AIDS after an average of 10 years, we should be seeing more cases, not fewer, given the estimated 32,500 HIV-infected people in this country. If this trend continues, a significant number of HIV-positive people will simply never get sick, implying that other factors may be necessary in order for an HIV-positive person to progress to full-blown AIDS.
So, of course, seroconversions are incredibly varied. That's why the low-risk seroincident people are so intriguing, the ones identified more recently in the studies with 20 to 30 years mean time free of AIDS. I mean, how can we explain it if it's so deadly?
And even Luc Montagnier has his doubts.
No, I think it's important to tell the truth to people. And HIV infection is like a Damocles sword. And of course, it's not a condemnation to death. I think it's important to that people which have infected realize they have some hopes, and the more time is passing, the hopes is increasing.
If you look at the total number of people with HIV antibody in Canada and the total number of deaths, it's still going to take 125 years or 150 years to kill everybody. In the hemophiliacs, it's going to take 200 years. So it's not adding up. It's falling flat on its stuff.
Canadian government statistics divide the total number of AIDS cases into those who are alive and those who've died. As time has gone on, the proportion of AIDS deaths has steadily increased as fewer and fewer HIV-positive people enter into the AIDS category.
So Peter is quite right there, Peter Duesberg. An HIV antibody positive test is not a death sentence, and clearly the epidemiology is pointing, thank goodness, sharply in that direction, which should be a sigh of relief for the advocates of patients, the people who work so hard like yourself and myself and the gay community to get a more honest, open view, and dozens of others. It should be a sigh of relief that the epidemiology is supporting the fact that HIV cannot cause AIDS in all persons who have antibody to it.
If HIV is indeed incapable of causing AIDS in and of itself, it will mean a major new research effort will have to be mounted to uncover the true cause or causes of AIDS. That's an effort that was suspended when HIV was announced to be the cause in 1984. But as Dr. Joseph Sonnabend points out, the sole focus on HIV has left many areas of pathogenesis unexplored.
Amongst the things that we didn't study were the role of herpes viruses, particularly of cytomegalovirus and reactivated Epstein-Barr virus infections in people with AIDS. Already in the early 1980s, it had been shown that cytomegalovirus infections were highly prevalent in people with AIDS, that these were not just your usual kinds of cytomegalovirus infections, but there was the infections with cytomegalovirus in people at risk, particularly homosexual men, was repeated. It was massive. It was known that cytomegalovirus could have immunosuppressive consequences, and therefore the role of cytomegalovirus in contributing to the immune suppression of AIDS, even if it's not saying it's a cause, but a virus that can produce changes in T cell subsets, produce some of the consequences that are seen in AIDS, such as overstimulation of B cells, are known to be produced by cytomegalovirus, but none of this was explored. So if cytomegalovirus contributes to the pathogenesis of AIDS, we've advanced nowhere, and there's good reason to think that it does. And there was good reason to think in 1981 that it did, and that exists today too. Epstein-Barr virus is the virus that causes mononucleosis. Reactivated Epstein-Barr viruses are frequent in people with AIDS, and some of the features of AIDS, such as an overproduction of antibodies by B cells, could readily be a consequence of reactivated B cells, reactivated Epstein-Barr viruses, infections turning on, infecting B cells and turning these B cells on to make immunoglobulins. That's totally unexplored. Modestly, little bits and pieces. I have been involved in some of the EBV Epstein-Barr virus work, and there's certainly, it's important to pursue this, but absolutely no interest in this whatsoever. There are other things. Some of the abnormalities found in AIDS were found early. I mean, they're not in 1981, 1982. What is the role of circulating immune complexes? People with AIDS have loads of circulating immune complexes. Now, even if they contribute something, if we had worked this out, we might find ways to remove them, ameliorate the condition of people. We've done absolutely nothing along these lines. Autoimmunity, we found, the group I was working with in 1982, that people with AIDS had autoimmune phenomena. Nowadays, there's a kind of interest in autoimmunity to try and explain how HIV causes T-cell death, but the autoimmunity that we're speaking about here is an autoimmunity directed against not just lymphocytes. There are a whole variety of auto-antibodies, antibodies directed against the body that appear in people with AIDS. Nothing to do with HIV, but they're there. Do we know whether they contribute to disease or not? We haven't pursued this at all. And if they do, we might have found ways to at least ameliorate that aspect of it. Interferon, we found interferon in the blood of people with AIDS in 1981, the very year that AIDS was discovered. It's not physiologic for people to be exposed to the high levels of interferon in a chronic fashion. It's seen in a disease like lupus, and in fact, its appearance in lupus, the fact that it appears in lupus, which is a classic autoimmune disease, when we found the same kind of interferon in people with AIDS in 1981, people I had worked with had also described it in lupus just only a year or so before that. That led us to look for autoimmunity in AIDS. It was rational to do so. If interferon in high levels appears in the blood of people with an autoimmune disease, if the same kind of interferon appears in the blood of people with AIDS, then it's reasonable to see whether or not AIDS is also has autoimmune components, and that's how we came to find that. So now, to this day, we don't know what the stimulus to make interferon is. We don't know what cells make it. This is virtually 10 years since its first discovery. Almost nothing has been done to elucidate whether or not interferon is contributing to the immune deficiency, because there are lots of theoretical reasons to believe that it might indeed be doing so, and so we haven't even begun to think of ways to determine this and then find out ways to try and control the abnormal production of interferon. We have absolutely done nothing. We've known about it since 1981, so those are some aspects of pathogenesis that have been totally neglected.
We do not know what causes AIDS. And as long as we do not know what causes AIDS, it should be an open field. And any remotely reasonable hypothesis should be eagerly investigated. AIDS is killing people. And when you have a situation of this magnitude, you really should be humble and listen to anybody with half a brain.